Yes, so this work was done in BRCA-associated triple-negative breast cancer, and it investigated the immunomodulatory effects of PARP inhibition. And we found that PARP inhibitors induce an anti-tumour immune response in BRCA-associated triple-negative breast cancer, but not in BRCA-proficient triple-negative breast cancer. We also found that this T-cell recruitment is mediated through activation of the SIGA-STING pathway within cancer cells, and that signals in a paracrine manner to antigen-presenting cells. And we finally showed that activation of the STING pathway within cancer cells is both necessary and sufficient for the anti-tumor immune response to PARP inhibitors and the therapeutic efficacy. We utilized a murine model of BRCA-associated triple-negative breast cancer. We evaluated the efficacy and immunomodulatory effects of PARP inhibitors using flow cytometry, immunohistochemical analysis, and immunofluorescence microscopy. And we also performed work in human triple-negative breast cancer cells, as well as isogenic pairs of BRCA-deficient and BRCA-proficient to allow a head-to-head comparison of the role of BRCA in this process. So this study takes a novel approach of depleting STING within cancer cells, and that completely abolished T-cell recruitment in response to PARP inhibition. And this implies that activation of this pathway exclusively within cancer cells is both necessary and sufficient to elicit an anti-tumor immune response, and that is critical for the efficacy of PARP inhibitors. Our findings suggest that in addition to combining PARP inhibitors with immune checkpoint blockade, BRCA-associated triple-negative breast cancer patients could benefit from a combination of PARP inhibitors with STING agonists that are already being evaluated in clinical trials.